All right. Good evening, everyone. Good evening. Okay. So, it was estimated that the exam will happen, they have said that, between 25th to 27th July. Okay. So meanwhile, we will focus on all the PYQs at least from 9 to 10 units completely. Okay. So we'll focus on the PYQs of 9 to 10 units. Especially we will focus on 2023 December paper to the 2017 December paper. How many questions that we can do, we can focus on. We will complete what we can say one unit in 2 days. Okay, because the units are very bigger. It takes minimum of 2 days time. All right, now let's continue. Okay, don't be hurry. Every one of you, don't be confused also. Always think and give the answer. Okay, and at the same time, don't take too much of time to answer the simple questions also. So because we have to cover maximum number of questions, so I'm requesting all of you, to do little bit faster. All right, look into the question number 1. The figure below depicts the absorption. We'll start with the 2023 paper. The figure below depicts the absorption spectra of chlorophylls and carotenoid over a range of the wavelengths. And the question says that which of the following combination best describes A, B, and C from the absorption spectra shown above? It's a very simple, straightforward question only. The question says that which one of the following combinations best describes A, B, and C? Okay. What about others? Every one of you, right or wrong, don't worry about that. Give the answer. We can see very clearly here. You can see, right? This will be the chlorophyll B. Correct, na? And this will be the chlorophyll A. And C will be the carotenoid. So anyway, you can eliminate the option 2 also, option 4 also very easily. And the correct answer will be the option 3. Chlorophyll B, chlorophyll A, as well as the carotenoid. No point of getting confused. It's easy to understand also. All right, everyone, let's look into the next question. The following are the certain statements regarding the PS2 electron carrier during the light reaction of the photosynthesis. Every one of you. Okay, every one of you slowly think and answer. And the question says that which is the correct statement or statements. Obviously, if you look into the question number 2, answer will be the 3. Good, good, good. The first electron released from the reaction center P680, it is transferred to the plastoquinone A to produce the plastosemiquinone. And why option B is wrong? Actually, QB is the mobile plastoquinone. But they said that the QA is the mobile plastoquinone. This option is the wrong option. No point of getting confused. The first electron accepted from Q to QB converts QB into plastosemiquinone. That is also wrong. QB is tightly bound to the complex. That is also wrong because QB is the major mobile one. Hence, the correct answer is the third one. Good. Think properly and answer the question number 3. The question number 3 says that which one of the following statements regarding the pep carboxylase is incorrect? Everyone, please look into the given question, friends. They are saying that which is incorrect. Okay, look into that incorrect. I believe you remember this. So here says that option A, during the day, they are saying that which is incorrect. During the day, C4 pep carboxylase is inactive and where CAM pep carboxylase is active. All the time remember the common point. CAM pep carboxylase enzyme always active only during the night time, and it will be inactive during the daytime. Correct na? But they have given the opposite statement here. Whereas during the day, C4 pep carboxylase should be absolutely active. Why? Because it has to combine the phosphoenol pyruvate with the carbon dioxide, and it is going to give rise to the oxaloacetic acid. So it should be active compulsory, but they have given the statement as the wrong. So hence the correct answer will be the number 1. Pep carboxylase is inactivated by the dephosphorylation. This statement is a true statement. Pep carboxylase kinase is the enzyme which is phosphorylating the pep carboxylase. That statement is true. The synthesis of pep carboxylase kinase is modulated by the circadian rhythm. That statement is also absolutely true statement. No point of confusing over there. All right. Now, every one of you look into the question number 4 and go a little bit faster. Rubisco enzyme is involved in both reductive and oxidative carbon cycle in plants. Following are the certain statements regarding them. You can easily choose the answer, but think properly and choose the answer. No point of hurrying. And please don't think that you have to answer every single question correctly. Okay, don't do that. Whichever you can guess, guess it perfectly. That's enough. Okay, what about others? Everyone. See, friends, we all people can, understand it so clearly, friends. It's not at all difficult question at all. We all people know that option A says that sugars are produced in both cycle. That is absolutely wrong. So sugar, I have already told you multiple times while giving the definition of the photosynthesis only, I told you multiple times. Photosynthesis is a process where there is a reduction of the carbon into glucose. So what is the point over there? So sugar is actually producing only in the reductive cycle. It never produced in the oxidation cycle. You can eliminate the option A. If you comes to know that option A is wrong, you can eliminate the option 1 also, option 2 also. And you see here, NADP and ATP are used in both cycle. That is also absolutely wrong statement. Why it is absolutely wrong statement? Because NADP and ATP, they are produced in the light reactions, but they are used in the dark reaction. Correct, na? But they are saying that they are used in both cycle. You know, you can't. They will be used only in the reductive cycle that they produced in the oxidation cycle. Correct or not? Hence, this is also wrong answer. Hence, immediately you can come to a conclusion that answer 4 will be the, number 4 will be the correct answer. No point of confusing at all. All right, next one. Question number 5. Easily eliminate this also. Very easily you can eliminate. This is there in the slide itself in a diagram. Simple question it is. And don't worry if you don't know the answer, but try, give a guess. If you are unable to guess 100%, just leave the question, no problem at all. No, so they look into this, everyone perfectly. Is it 4? I don't think so. Check it once again. Okay, every one of you look into this. There are 2 type of enzymes are there. Okay, this is the enzyme 1 and This will be the enzyme 2. Every one of you read the questions once again. Option A says that sucrose phosphate synthetase enzyme. This is one enzyme. Phosphate synthetase. Then another one is called as sucrose phosphate phosphatase enzyme. No point of getting confused. One is the synthase enzyme. One is the phosphatase enzyme. Now, you tell me, Shafia, tell me, Swati, what is the function of a phosphatase enzyme? Tell me. What is the function of phosphatase enzyme? Phosphatase is the enzyme which is actually remove the phosphate group. Yes or no? If it wants to remove the phosphate group, that specific substrate should have the phosphate group. Correct or not? Very good. That substrate should have the phosphate group or not. Now you see the option B. Option B says that fructose 6-phosphate and UDP glucose are the substrate of the SPP enzyme, which is absolutely wrong statement. So SPP enzyme cannot remove the phosphate group from the glucose. It remove the phosphate group only from the fructose 6-phosphate. Option B is actually a wrong option. There itself you can come to a conclusion that the answer will be the option 3. Don't get confused. Fructose 6-phosphate is one of the substrate for SPS enzyme, right? Sucrose is the final product of the SPP enzyme. Of course, that is also right. UDP is one of the product of the SPS enzyme, while PA is one of the products of SPP enzymes. This is also true. Hence, the correct answer will be the number 3. It is there in the slide of the photosynthesis topic. I want you to read it once again. Okay. Whatever your guess is also valid guess, but look into the point very correctly. Now, during glycolysis in plants, alanine and related amino acids are directly produced from which one of the following precursors? Question number 6. Quickly, little bit quickly. Little bit quick. So we all commonly know that alanine is what? Alanine is basically an amino acid. Amino acid is basically going to give rise to what? It is going to give rise to the pyruvate. We commonly know that. All right, all right, all right. Every one of you think properly and answer the question. Question number 7. Question number 7, everyone think slowly and give the answer. The following statements are made regarding the nitrogenase enzyme involved in the reduction of the atmospheric nitrogen to ammonia. Simple question it is also. Anyone else? All right. See, nitrogenase enzyme composed of a two components. One is called as what? Dinitrogenase and dinitrogenase reductase. Option A is absolutely correct. If option A is correct, you can eliminate the option 2 also. You can eliminate the option 3 also. Correct, na? And you also know very well, suppose this is the enzyme. Here, iron sulfur cluster is present. Here, molybdenum iron cluster is present. And we all people commonly know that here, nitrogen will actually come and attaches. So this is also a true statement. Iron protein first, electron reaches to the iron protein to the molybdenum iron to the nitrogen. And we also studied that iron sulfur protein associated with what? Hydrolysis of ATP. That is also right. Now, molybdenum iron protein component is a homodimer. It is a wrong statement. It is a heterodimer. Iron protein component is the dinitrogenase. No, that is also wrong. It is the dinitrogenase reductase. Hence, the correct answer will be the number 4. Direct question it is also. All right. Think slowly and analyze the question properly and give the answer. The following are the statements are made regarding the cytokinin biosynthesis in the plants. Slowly, slowly. Don't give me too fast answers. At the same time, not too late. They're asking correct statements. Except this one, you have read everything. In the slides itself, it is there. The following statements are made regarding the cytokine biosynthesis in plants. Okay. Option A says that transgeatin and isopentenyl adenyl IP are the common what we can say active forms of the isoprenoid cytokinins. That is what we can say that true statement. Option B, CKs are present as a nucleoside and glycoside conjugates, but not as a nucleotide conjugate. That is wrong statement, right? Cytokinins compulsory present in the nucleotide analogs also, right? So option B is wrong. If we comes to know that the option B is wrong, you can eliminate these three options very easily. Then you can find out answer will be the three. Anyway, dephosphorylation, deribosylation. I believe you all people remember this is there in the slide itself. First, we remove the phosphate group. Later, we remove the sugar. I have explained you in the biosynthetic pathway very clearly in the slide. Are the two steps involved in the cytokinin formation? This is right. So remember, log is the enzyme which is involved for the final biosynthesis of the gelatin. Okay, that is also right. Hence, the correct answer will be the option 3. It is there directly in the question itself, slide itself. Cytokinin metabolism slide. Look into that very carefully and answer. Next, question number 9. Which one of the following statements about the phytohormone ABA is correct? Again, friends, they're asking the question which is correct. They're asking correct. Tell me, Kiritika, what is the answer for this? Which one of the following statement about phytohormone ABA is correct? Try it, try it. Everyone try it. They're asking, what we can say. Correct. Every one of you answer the question. Abscisic acid inhibit the vivipary or abscisic acid promote the vivipary? Every student please answer. Abscisic acid promote the vivipary or inhibit the vivipary? Of course, it is inhibits. And ABA BD glucosyl ester is an active form of the ABI. No, it will be an inactive form of the ABI. And inactivation of ABA involves its oxidation to phasic acid. Absolutely right. Right. Now all the students answer abscisic acid biosynthesis occur entirely in the plastid. Everyone tell me where the abscisic acid biosynthesis occurs. Every student please answer. We read it very clearly where the abscisic acid biosynthesis generally occurs. Very good. We generally says that abscisic acid is a kind of a ubiquitous plant hormone. So it is at a lower concentration. I'm using the word lower. At a lower concentration, it is getting biosynthesized at the every part of the plant body, but at very lower concentration. No point of getting confused. Which one of the following plant-derived molecules are widely used as an analgesic? Don't worry, don't think that you have to answer every question. Whatever you are trying to answer, make sure that it is correct. And we all people read, right? Salicylic acid. I hope you all people remember we generally use the drug aspirin. Aspirin basically contain what? Salicylic acid only. What it is act like? It is acting like, powerful analgesic, which relieves us from the pain. Very good. Okay. No need to get confused. It's a 4-marker question, slow, so easily you can answer. Given below are the 5 experiments A to E showing the effects of the duration of the light and dark periods on flowering of the short day plants and long day plants. And the question says that which are correct. The question is asking which are correct statement. They are saying correct. Oh, sorry, sir. You can easily eliminate option. Please use elimination. Only use elimination. So fast. Okay, what about others? Safia said 2. Others? Akila is 1. Okay, others. Slowly think and analyze. Swati said 3. Okay. All the students, anyone else give the answer? All of you look into this, friends. It's so easy. Why you are feeling difficulty? Look into the given question. Okay. Look into this. This is the light treatment, flowering response. They have given short day plant. They have given the long day plant. First of all, every student tell me, in the you all people know very well, when the dark is more, undoubtedly it will favorable short day plant. You know it very well. At the same time, you also know very well, when the light is more, long day plants will flower. The point is very clear, no confusion at all. So in the first condition, which is high, dark is very high, undoubtedly which will flower, short day plant will flower, no doubt about it. And since the night length is very high, long day plant never ever flower, it will be vegetative. Absolutely true statement. No point of getting confused over there. Next one, very important one. Option B, tell me which is more. Daylight is more. When the daylight is more, then obviously which will flower? Long day plant will flower and short day plant never flower. Here itself you will come to know, friends, option A is correct option, option B is also correct option. So you can eliminate the option 3 also, you can eliminate the option 4 also very easily. Now, look into the option C. All the students, first don't get confused, friends. First tell me, which is originally more? Dark is originally more. Then which has to flower? Dark is originally more. Which has to flower? Short day plant wants to flower. But what we have done, we have exposed the light. We have exposed the light. But when we expose the light, which length will increase, as friends? Day length will increase. When the day length is increasing, which plants has to flower? Long day plant has to flower. But they said that long day plant is not flowering, short day plant is flowering. There itself you can eliminate option C is wrong, and you can come to a conclusion that answer 2 will be the correct. Correct or not? Option U, if you look into this, daylight is little bit higher, then obviously there is a flowering of the long day plants. Easily we can come to the conclusion answer will be the option 2. No point of getting confused over there, and it is very easy to understand. Good. Okay. Question number 12. Which one of the following statement is not a characteristic feature of the aquaporins? They're asking not. See, aquaporins are actually present in the bacteria also. Undoubtedly, aquaporin is what? Aquaporin is an integral transmembrane protein generally. Correct, na? So they are integral membrane proteins in the major intrinsic protein family. That is right. Right. They are not absent in bacteria. Actually, they are present. A highly conserved NPS sequence present at the N terminal also, present at the C terminal also. No point of getting confused over there. Hence, the correct answer will be the 2. Good. Take your time. Slowly answer the question. following statements are made regarding the plant natural products of terpenes? Eliminate, always eliminate. Okay. Others. Okay. You all people know, friends, monoterpenes are how many carbon containing compounds? Every one of you tell me monoterpenes are how many? Monoterpenes are 10 carbon. Correct, na? There is something called as a hemiterpene. Hemiterpene is generally called as the 5 carbon. Monoterpene is the 10 carbon containing compound. There's no doubt about it. This is a true statement. Once we comes to know that the A is the true statement, you can eliminate the option 2 also. You can eliminate the... Okay. Here they're asking what? They're asking the true statement, right? So option A is a wrong option. If you comes to know that option A is a wrong option, you can eliminate option 1. You can eliminate the option 3 also. Correct, na? So now, the next, what is the option B says that anti-malarial drug artemisinin is a sesquiterpene. That is true, which is a 15 carbon containing compound. These are all there in the slide itself. So option B is the right answer. Azadirachtin, we studied that, which is present in the neem tree, which is fighting against more than 25,000 different type of the insects that are present in the plant. Right. Taxol is basically acting as what? A specific anti-cancer drug. That is also right. Hence, the correct answer is number 4. No point of getting confused over there. Right. Take your time, slowly analyze and give the answer. Given below are the list of the plant-derived alkaloids and their use in the modern medicine. I use it to explain you this table every single time. Easily you can come to the conclusion. All right. Anyone else? All right. No problem. Look into this. Caffeine. Right? Why we take the caffeine? Caffeine generally stimulate the central nervous system. That's why I told you multiple times. I use it to give the same example to all of you. When we drink the coffee, once in the span of 2 to 3 hours, it will make the central nervous system stimulated. But if you're drinking continuously, it leads to the depression. So caffeine is powerful central nervous system stimulant. Just now we discussed morphine is the powerful narcotic analgesics which causes the hallucinations and delusions. And in the previous question also we have seen that quinine is the anti-malarial drug. Vincristine, and remember another example, vincristine and vinblastin. Vincristine and vinblastin, these two are what, friends? Generally vinca alkaloids. Vincristine and vinblastin are vinca alkaloids. They are actually acting they are acting as an anti-neoplastic drugs. No point of getting confused over there. Hence, the correct answer will be the number 4. It's a very clear-cut point. Okay. See, every question is there from the slide itself. Okay. I believe you all people answer. During the water stress, ABA increases dramatically in leaves, causing the stomatal closure. Given below are the various events that are involved in the process. And you have to choose the correct sequences. You have to choose the correct sequence. Slowly, every one of you. No, I have explained it to everyone, this diagram itself. Everyone forgot. No, we have seen this. How come you people forgot this? Look into that correctly, every one of you. Mm, look into that. All right. Anyway, I hope you all remember, friends. See, I never encourage any one of you choosing the wrong answer from the concepts which you read multiple times. This is just you are confusing yourself and you're choosing the wrong answer for a simple question. And anyway, we studied very clearly that ROS pathway. Let me show you that. What is the another pathway? IP3CADPR pathway. Correct na? Cyclic ADP ribosylated pathway. I hope you all people remember that. Now, first, in this particular pathway, what is the first event that will happen? We all know very well the first event is abscisic acid will come and attaches. Correct or not? One second, just give me one moment. I don't know why it is hanging. Just a moment enough. Okay. All right. We all studied it multiple times. Okay. All the students focus on the picture correctly. First, what happened? You can see the picture. Abscisic acid will come and attaches first. When abscisic acid comes and attaches, see this, I have written it so clearly. See, every one of you look into the picture also correctly. Abscisic acid attaches. ROS IP3 pathway will be activated. Look into this. First, calcium channels will be opened. Every one of you look into this. Once the calcium channels will open, you can see that then the potassium influx, potassium efflux will happen. Potassium influx will be blocked. Correct, na? Very straightforward question only. So first is binding of abscisic acid. C is the number one. Later, activation of the ROS pathway. D is number two. Then what? You have to remember first calcium channels will be opened. After the calcium channels only, the potassium channels will be closed. So the answer will be the option 4. No problem. Slowly think and answer the question. No problem. Slowly. Okay. First answer this matching question. What is that? Slowly choose it. This is also I have explained it. Oh, sorry. Easily you can choose the answer. Even though you know one option, you can choose the correct answer. All right. What about others? No, we all people studied multiple times, friends. Even in the chapter of the photosynthesis also, we all people studied, friends. When the light intensity is higher, what do we call that? When the light intensity is higher, all the photosystems, all the electron transport chain is getting inhibited. So what do we call it as? That mechanism is called as a photo inhibition. You all people very commonly know about that. Correct, na? And when you look into the flooding, friends, actually what will happen in the flooding? Obviously, there is an extremely low supply of the oxygen to the roots. Very good. Then if you look into the salinity, ion toxicity and water deficit, this is also you people can answer so directly. Water potential reduction. Hence, the correct answer will be the option 2. And when you look into this, the question number 18, if you read about systemic acquired resistance, then only you can answer the question. If you did not read it, you can't answer the question. Okay, it's a very simple. And if you know it only, you people try to answer. The following statement refers to the observation made by student upon using 2,6 difluoro isonicotinic acid to induce systemic acquired resistance in tobacco. You people answer some questions with common sense also. They are saying that they have used an inhibitor. Whenever they used an inhibitor, and what the option A says that upon using the inhibitor, they're saying that salicylic acid concentration got enhanced in the plant. No, actually, I want you to understand the point. 2,6 dichloroisonicotinic acid, which inhibit the action of the salicylic acid. So option A is a wrong option. It never enhances the activity of the salicylic acid. Slowly. Just now I'm telling you, right? Okay. Slowly analyze. Then it cannot enhance us. So option 1 is wrong. Option 2 is also wrong. Right. Then it does not enhance salicylic acid concentration in the plant. Obviously, this statement is the true statement. Now, no point of getting confused here. Option C actually says that fails to activate salicylic acid resistance in NAHG expressing in plants. I want all the students has to understand my words very correctly. Whenever an inhibitor showing its action on the salicylic acid or in that particular plant. First, if we want to allow the salicylic acid to be expressed, it is very important we have to activate all the genes that are required for the expression of the salicylic acid. Among many genes that are involved in the expression of the salicylic acid, one of the gene is the NAHG gene or NAH gene, we generally call it as. If it is inactivated, SAR cannot be activated actually. So if an inhibitor is acting on the tobacco plant, first there is an activation of this gene, then this gene will allow the activity of this particular salicylic acid. But since the inhibitor is present, the salicylic acid action also cannot be shown. There is no point of getting confused with the option C and option D. All of you try to understand the point correctly. So there is 2,6 dichloroisonicotinic acid. What is their function? No point of getting confused. Whenever this particular inhibitor acting on the tobacco plant, first what will happen, whenever it is acting on the tobacco plant, immediately, see, since there is an inhibitor entered into the plant, immediately the NATH gene will be expressed. No point of getting confused, friends. Whenever the NATH gene will be expressed, it is going to release what? It is going to release the salicylic acid. But what is actually happen? Even though the gene got activated, even though salicylic acid released, the inhibitor will come and inhibit the action of the salicylic acid. So the point is what here? The gene expression will occur. So that gene will allow the synthesis of the salicylic acid also. But the salicylic acid concentration will be inhibited because of the activity of the inhibitor. So B is right answer as well as the D is also right answer. Don't get confused here. Here gene expression occur, salicylic acid synthesized, but the salicylic acid activity cannot be shown. No point of confusion. Answer is the number 4. Slowly think and answer. Now, this is a very common question that they use to ask in the exam. Very common question. That is the pathogenesis related proteins. See, you people answered more than 85% of all the questions correctly. That's okay. If you did not answer some questions, don't touch that. And friends, 1 second. Whenever it comes to this question here, nothing to explain, friends. You people has to remember this correctly. Okay. PR2 beta 1,3 glucanase, correct na? And PR3 is the chitinase, whereas PR6 is proteinase inhibitor. PR10 is the ribonuclease-like structure. That is true. That is very true. Answer will be the number 2. Very good. Very good. Okay. Which of the following leads to the induction of the defensin PDF 1.2 in the Arabidopsis thaliana? Try it. See, okay, I'm just giving you, friends. There are two major wounding hormones are there in the plant. Major, I'm talking major. Among those, first major wounding hormone is the ethylene. Then the second wounding hormone will be the auxin. Okay. Then third will be the jasmonic acid. These three are the major. But the point is here, auxin can be released only whenever the vascular tissues got damaged, such as xylem got damaged, phloem got damaged. Only at that specific condition only they are going to release. Correct, na? But the major wounding hormone will be the ethylene. So answer is 4. All right, very good. Most of the people has answered more than 85% of the questions very correctly in the 2023 paper. That's really good. All right. 2022. Look into this. Which one of the following statements regarding the chlorophyll is not correct? They're saying not correct. All right. What is the answer? Oh, sorry. The no chlorophyll A we already studied, right? Every chlorophyll molecule generally having porphyrin head and phytol chain. Porphyrin head and phytol chain. No point of getting confused. Chlorophyll A having what group? Methyl group in its side chain. Chlorophyll B having aldehyde group in its side chain. No point of getting confused over there. Now, option 3 says that only chlorophyll A, but not chlorophyll B as the magnesium. Every chlorophyll A, B generally having a cofactor, sorry, not cofactor, what do we call magnesium as the chromophore, which is located in the head of the porphyrin. So this statement is the wrong. The long hydrocarbon tail, the common statement, it embed the head on the plasma on the thylakoid membrane. This is the right statement. The only thing wrong is the option 3. Good, good, perfect. Question number 23. During light reaction in photosynthesis, electron is transported in electron transport chain and produces ATP and NADPH in the process. Following are the certain statements regarding the ETC during the light reactions. We can normally answer the question. Electrons from P680 moves to the first to quinone and then to the pheophytin, which is a wrong statement. So first, from the P680, the electron is generally moving to where? It is moving to the pheophytin. Pheophytin later quinone. So option A itself wrong. Then only we can come to that option 1, 2, 3 are the wrong options. There itself we can choose the answer as the option 4. Okay. Identify the correct site of action of DBMIB 2,5 dibromo 3 methyl 6 isopropyl P benzoquinone, an inhibitor of the electron transport chain. This is, it's not any kind of analytical or those kind of question. If you know the answer, you can answer. If you don't know the answer, you cannot. So plastoquinone A, plastoquinone B. Later, there is a cytochrome B6F complex. Exactly here, the electron flow can be blocked by this particular benzoquinone. Answer will be the 3. Simple. Let me write option 3 here. Option 3 will be D, option B is C and D. Option D will be the A and D. All right. So every one of you focus on the question. A student listed following combination of enzymes and their involvement in different phases of the Calvin-Benson cycle. You have to choose the combination of correct statements. See, if you miss the enzymes of the Calvin cycle, this is what the point. You have to remember the enzymes. At least if you remember one also, you can answer the question. Actually, in the regeneration phase, friends, what we are producing, tell me, every one of you. In the regeneration phase, what we are producing is the RUBP. Correct or not? When we produce the RUBP, we produce the RUBP with the help of a phosphoribullokinase in the regeneration phase. Option D is absolutely wrong. Sorry, absolutely right. Then you can eliminate option 1 and 2. So this is very, very clear. Now, if you look at the option A, phosphoglycerate kinase, what is the function of this particular phosphoglycerate kinase? It is converting 3-PGA into G3P. Glyceraldehyde 3-phosphate can be formed by the phosphoglycerate kinase enzyme, which is in the reduction phase. So A is correct, D is also correct. There itself you can come to the conclusion. Glyceraldehyde 3-phosphate dehydrogenase, there is no use of the regeneration. And we don't produce the triase phosphate in the reduction phase. No, it will be in the only fixation phase. Hence, the correct answer is option D. No point of confusing at all. Very simple questions. Photochemically generated ATP is consumed in which one of the following phases of the Calvin-Benson cycle? Slowly, slowly. Photochemically generated ATP is consumed in which one of the following phases of the Calvin cycle? So we know, right? Reduction and regeneration. Reduction, we use the 6 ATP. Regeneration, we use the 3 ATP. So only in the reduction and regeneration only we are going to produce the using the ATP. Now again the direct question. Which one of the following are the correct encoding sites of the large and small units of the Rubisco in the red and brown algae? See friends, they are not at all asking in the plant. No, just now I am telling you the same thing. They are not asking in the plant. You're not looking into the given question. They are not looking into the plant, asking the plant. If it is a plant, large subunit is in the chloroplast, small subunit is in the nucleus. If it is a plant, correct na? If you look into the algae, every one of you remember, all will be present in the chloroplast only. No point of getting confused over there. Answer will be the 4. Don't confuse. All right. Question number, what we can say, 28. See this question in the 2023 question paper, they asked for 4 marker. Same question with a little modification in the 2022, they asked for the part B. Which one of the following correctly stated the action of the sucrose phosphatase synthase enzyme? They're asking synthase enzyme. Wonderful. Very, very good. UDP glucose and fructose 6-phosphate both can be used as the substrate for the sucrose phosphate synthase enzyme. Very, very good. Because friends, it is so simple, right? Glucose and fructose both are required for the formation of the sucrose. Yes or no? So simple it is. Glucose is required. Fructose is also required. Then only you can get the sucrose. What is the enzyme that is involved in the synthesis of the sucrose? Sucrose phosphate synthase enzymes, so both can act as a substrate. In the previous question, what they have given? They said that the glucose and fructose 6-phosphate are the substrate for the phosphatase enzyme. That is wrong statement. Option 1 is absolutely true. They are not the products. They are the substrate. Sucrose is formed as a product. There is no doubt about it. But this is not the more appropriate statement for this given question. Then sucrose 6-phosphate, no, they are not the products. Obviously, what is the answer? Answer will be the number 1. All right, think slowly and answer. I'll write it here. Option 3, B, C, D. Option 4, A, C, and D. All right. Following are the certain statements in the photorespiration pathway in plants. So fast. Every one of you slowly think and answer the given question. Some people said 1, 3, everyone. Very good. Okay. What is the answer? What is the answer? No, this is a kind of a difficult, confusing question. First, question says that which is incorrect. First point. First point is which is incorrect. No, friends, you are not getting the point. Which is incorrect, they are asking. Now, all the students answer my question. A is right or wrong statement? Tell me, Selvi, A is the right statement or wrong? No, you people are not getting the question. They're asking which is incorrect. A is the right statement. A is the incorrect statement. No doubt about it. Correct, na? A is the incorrect statement. Very good. And what about option B? Glycolate exit the chloroplast and enter into the peroxisome. Is it the true statement or false statement? Option B. It is a true statement because glycolate entered into the peroxisome. Very good. There only it is converted into glycine. So option B is correct. Option A is wrong. Glycolate that is synthesized during the C2 cycle enters the chloroplast from mitochondria. Is this statement is a true or false? Is this statement is true or false? From where to where it entered? It entered from chloroplast to peroxisome. Correct, na? So this is a wrong statement. So A is wrong, C is also wrong. Now, tell me why the option D is wrong. I am not getting why the option D is wrong. Glycine is transported from peroxisome to mitochondria. There only glycine is going to be converted into serine. So why option D is a wrong option? I am not getting. Why option D is wrong? Option D is right. Hence the correct answer is number 1 here, which is incorrect. Correct or not? Glycine synthesized in the peroxisome. From the peroxisome, it enter into the mitochondria. In the mitochondria, it is converted into the serine. Correct or not? Yes. Hence, the correct answer will be the option 2. Slowly, slowly. Should not make small questions wrong. Okay, no problem, no problem. Okay, we already studied the same thing in the biochemistry also. Glycolysis and citric acid cycle contribute precursors to many biosynthetic pathways. Column X lists name of the precursor and column Y lists the product synthesized. Slowly. Oh, this much fast. All right. Hexose phosphate, cellulose, pyruvate, alanine. Then what about that? Pentose phosphate, nucleotide, and oxaloacetate, aspartate. Very good, very good, very good, very good. Answer will be the two. Very good. Okay. Question number 31. And please look into the given question very carefully, friends. The question says that arrange the above pathways from the lowest to the highest. Friends, don't get confused. They're asking lowest to the highest. Lowest they're asking. Others, question number 31. I have already told you in the first slide itself. You can fix the nitrogen industrially. Natural lightning can fix the nitrogen. And we also studied the biological nitrogen fixation. Correct or not? Undoubtedly, which will be the lowest? Industrial nitrogen fixation is the lowest. Then followed by the natural terrestrial system. Correct na? Then followed by the industrial nitrogen fixation. Then finally, biological nitrogen fixation. Answer will be the 3. No point of confusion at all. Okay. No problem. You can answer this question. Which of the following domains is present in the symbiosis receptor like a kinase proteins? It is actually, the question of the mol bio. Even it is there in the biochemistry also. This we read in the eukaryotic gene regulation. Very good. So the common symbiotic receptor is LRR receptor. Leucine rich repeat. No point of getting confused over there. Okay. Try your best. Slowly analyze the given answer and answer. Slowly. Yeah. Plant nodulation gene encode proteins with receptor like features. Following are the list of some nodulation proteins and their possible domains. Just now you all people answered one question already. Same thing you can correlate. Just now you have answered leucine rich repeat. Just based on that you answer. So fast, everyone. No, dear Swati, that is LRP receptor. Okay, that is LRP. In the Wnt signal, we read LRP. That is different. Everyone, what's the answer? It's a simple. Just now you read, no? Symbiosis receptor kinase. It is basically what? LRR, leucine rich repeat domain. No point of getting confused over there. Then what is A? Every one of you. Entry receptor means what? All the students listen. Which is the extracellular domain. No point of getting confused over there. And what about the signaling receptor? It lacks the kinase features. That's all. So answer will be the number 2. Okay, this is A2, B3, C1. This will be the answer. It was not typed correctly. A2, B3, C1. Option 2 is the correct. Okay. Now, this is actually a biochemistry question. When we are doing the biochemistry question, there we will read it. Okay. Next, which of the following nitrogen compounds is formed during the deamination of the organic nitrogen in the plants? Deamination, removal of the amino group. Of course, friends, removal of the amine group, there is a formation of the NH4. See, NO2, NO3, how we can produce NO2, NO3? From the NH3 and NH4 only, we can produce NO2. Then followed by the NO3. So from the amino acid, if there is a deamination occur, then what we get? We get the NH4 only, right? Removal of the amino group, which is giving rise to the NH4. It's a simple one. Don't confuse. All right. The plant hormone gibberellins are the group of, you all people commonly know that, which is the diterpene. Leave it. 20 carbon. Okay. I want everyone of you try this question correctly. Question number 37. Following statements were made regarding gibberellins biosynthesis in the plant and fungi. What's the answer? Everyone has given the answer. And what is the question says that correct? Two separate enzymes are involved in the synthesis of the antaquarin from GGPP in the plants. Of course, true. There is no doubt about it. Only a single, what is the answer? Yeah, correct. Only a single bifunctional enzyme catalyzes the synthesis of antaquarin from the GGPP in the fungi. This is also right. GA biosynthesis generates mostly clustered on a single chromosome. Right. It never be randomly located. Hence, the correct answer is the number one. All right. Is it clear, everyone, so far? There are 12 more questions are there. I want you to solve that. Only 12 questions. Only 12 questions are there. You solve it. And in the tomorrow class, I will solve 2020 to 2017. By the plant physiology will be completed. Okay. I am sharing you right now all the questions with answers. And I want you to, without looking the answers and explanation, first answer the question. Okay. By tomorrow, I'll complete the 2020 to 27. So there are only 12 questions are left out. Please solve the 12 questions without fail. All right. I'll share the question paper, in a while. Okay. All right. Thank you.